So Kevin, I understand that you worked in a PhD program working with some medical devices, and I was wondering if you could please tell me a little bit about what your work was and what you did in that program.
Sure, I did a thesis using a biocomputation method to study cancer profiles. More specifically, we were hoping to see if we can find some biomarkers that will help us detect and diagnose cancers.
What exactly are biomarkers?
Biomarkers are features that you can use to detect or diagnose diseases such as cancer. So for example, you take a blood sample of somebody and then you can profile the gene expression of the cells in the blood. And you can see what genes are turned on or turned off in those blood cells. And you can compare the cancer cells with the non-cancer cells and see what are the features of cancer patients in their blood.
I gotcha. Is that something that is typically associated with the person's original DNA, or do the differences in those two DNAs come from mutations that occur as that person grows older?
Typically, they come from mutations, and also not just mutation of sequences, but changes in the gene expression profiles. So I understand you were talking about gene sequencing. So thanks to the Human Genome Project, all gene sequences are done, and now we know the gene sequences, but gene sequences alone does not predict all the disease prognosis. We need to know what genes are turned on or turned off in different cells.
Yeah, specifically in cancer cells. And once you have that information, once you know which genes are turned on or off through gene sequencing, what can you do about it then? What do you do after you have that information?
I want to clarify. So not by gene sequencing. Gene sequencing is just a DNA sequence.
Yeah.
But each gene is expressed in different number of copies in different cells. So in some cells, some genes are expressed more than normal. So maybe in the cancer cell, some genes are out of control, and these cells just keep dividing because there's no cell repair mechanism to help these cells anymore. So what we do is that we can identify these profiles, and we can take a blood sample, take Nick's blood sample, take anybody's blood sample, and see if we can find out what genes are being overexpressed. And maybe this will tell us if this person is at risk for cancer, or what kind of cancer this person might be at risk of.
I got it. Okay. When we had talked about gene sequencing, I understand that it was common for people to use a machine that was called a Selecta sequencing machine, in which they broke up the gene sequence into a number of pieces. They analyzed each of those pieces and then fit them together in a certain order. Is that the method that you guys used, or what method did you use to sequence a person's DNA?
So there's a public database now for all the gene sequencing, so we do not have to do that ourselves anymore. We can stand on the shoulders of a giant. So instead we use something called gene chips, or called cDNA microarrays. So these are gene chips that you can put our messenger RNA samples on and see what genes are being turned on, because we have these gene chips designed for each gene using the gene sequence from Celera and most more commonly from the public Human Genome Project database.
Wow, that's cool. Now, when you say we have these chips that are able to detect which genes are turned on or off, how many base pairs of DNA go on a single chip?
There are different types of gene chips. So the one that's the non-commercial one usually have longer sequences, say 100 or to 1,000 base pairs. For the commercial ones, the Affymetrix one, they are much shorter. I don't know how short they are, maybe between 20 and 100 base pairs.
Okay.
And these are just ballpark numbers. They probably change from generations of technology.
Wow. And so the gene chips, how do they sense? I imagine they sense what base pair, whether it's A, T, or C or G, they sense what base it is, and then they give you a readout as to which genes are turned on or off, and what that DNA will express?
So the kind of microarray we use just to allow hybridization between the messenger RNA sample or more specifically amplified version of messenger RNA sample to hybridize onto the DNA sequence on the gene chip. So we have a complementary sequence on the gene chip.
Okay.
And then you can use, because we're complementary sequence, they'll bind to each other, then we can see which ones bind to each other.
I gotcha. And by analyzing where they're bound, you can figure out what the sequence of it is.
By finding out how many sequences are bound to this gene sequence, we know how many copies they are, and then we know if turn on, turn off. So for a gene that's turned on, we'll have a lot of copies bound to this specific spot on the gene chip.
Okay.
So if a gene is turned off, then you will not find any sequence binding to this specific spot on the gene chip.
Okay. What exactly does it mean for a gene to be turned on or off?
So there are regulatory mechanisms in the cell to express specific genes under certain circumstances. For example, when a cell is dividing, you need genes that will be expressing proteins that will help with cell division and cell proliferation. So there are different genes involved in different processes in the cell.
I understand. And because of specialization of the cell, some genes get turned on or off based on what type of cell they're in. That's what you're saying. Okay.
So for example, the cancer cell is a cell that grows out of control. They keep growing, and there's no way to stop them. That's because it's cell proliferation or cell growth genes. They are turned on out of control. There's no mechanism to turn them off.
I gotcha.
So it's become tumor cells.
Yeah. Kevin, that's great. I sure appreciate you explaining these gene chips and everything. Thanks for your time.
Thank you.
Yeah.